There's something strange about a relationship that has no beginning you can point to, not in the way that childhood feels, that blurred edge where memory first starts and everything before it is just inference and photograph. This is older. This is a relationship so old that the bodies involved were still deciding what they were going to become. The nervous system that the parasite now uses didn't exist yet when the parasite arrived. The host was something simpler then, less defended, less wired for sensation, and the presence was already there. It's hard to find the right word for what a parasite is when you slow it down to this scale of time. Invader feels wrong somehow, too dramatic, too intentional. What it actually is, across deep evolutionary history, is something closer to a persistent biochemical neighbour, one that learned across millions of years of failure and occasional success which chemical doors to knock on. Most of them never opened. Those lineages ended. The ones we can study now, the ones inside the grasshopper, the ant, the snail, the rat, are here because they found doors that opened. Not on the first attempt, not on the 1000th. Across a span of time that has no good human analogy, something kept trying variations until a variation worked. That's not intelligence. It's just what survival looks like when you multiply it by enough generations. You carry a few assumptions about what it means to be a host without knowing you carry them. That the body is sovereign in some functional sense, that what you do originates in you, that the chemistry driving your behavior belongs to your biology, your history, your particular nervous system built across your particular life. Most of the time, for most organisms, this is essentially true. But there are exceptions so old and so refined that the body barely registers them as foreign anymore. The molecular signals involved have been shaped across so many host generations that they arrive quietly, find the right receptor, and proceed. No alarm, no inflammatory cascade, just a subtle shift in what the host's own chemistry does next. It's worth sitting with that for a moment. The shift doesn't announce itself. The earliest evidence of parasitic manipulation in the fossil record is locked in amber. An ant, approximately 48 million years old, biting down on a leaf vein with its mandibles locked in a grip that was never released. The fungus that caused this is identifiable from the bite pattern. The behavior it produced, that precise fatal grip at a precise height, was already fully formed 48 million years ago, which means the manipulation was already old then. The relationship between Ophiocordyceps fungi and carpenter ants almost certainly predates that amber specimen by a significant margin. What the fossil shows us is not an origin. It shows us a relationship already in its mature form, already running the same behavioral subroutine we observe in forests today. Whatever experiments produced this, whatever failed attempts, whatever partial manipulations that didn't quite work, those are lost to time entirely. We're looking at the refined version. There's something that stands out about this, and I'm not sure the word unsettling is quite right for it. It's more that it reframes something you might have taken as a given, that the host's behavior is the host's, that an animal's actions emerge from its own biology, its own adaptation, its own nervous system doing what it evolved to do. This is mostly true, but mostly is doing a lot of work in that sentence when you consider how long some of these parasitic relationships have been running. The carpenter ant that climbs has its own muscles, its own neurons. The leg movement is generated by the ant's own physiology. What the fungus has done, quietly at the cellular level, is alter what those neurons are told to do. The ant is not being moved from the outside. The instruction has been inserted on the inside. It's a distinction that feels important, even if what you're watching looks the same either way. What the host carries is no record of this, not in any accessible sense. The ant doesn't experience the manipulation as manipulation. The rat altered by Toxoplasma doesn't notice its fear response has been muted. The grasshopper walking toward water has no reference point for what it would have done otherwise. The absence of any felt difference, that's part of what makes this biology work. A manipulation that produced obvious distress would trigger immune responses, behavioral corrections, avoidance. What evolved instead are alterations subtle enough that the host continues functioning, moving, eating, sometimes reproducing, just redirected on a timeline that serves a different organism's survival. The body doesn't know it's been borrowed. These relationships exist across an enormous range of taxa: fungi and insects, protozoa and mammals, flatworms and fish, nematodes and crickets. They've evolved independently multiple times in lineages with no shared ancestor, which means the basic logic of behavioral manipulation isn't a single evolutionary accident. It's a solution that biology has arrived at again and again in different chemistries through different mechanisms toward the same general outcome. There is something in that convergence worth noticing. When the same strategy keeps emerging across unrelated organisms across deep time, it usually means the strategy is working against something stable, a biological constant, something about how nervous systems are built, how behavior is generated, how host defenses are organized, something that hasn't changed enough to close the door. The door is still there, the chemistry still works, you won't feel any of this happening. That's not meant as a reassurance or a warning. It's just what the biology produces, processes running beneath the threshold of sensation, beneath the reach of immune awareness in many cases, certainly beneath conscious access. The guest arrived long before memory did, long before the nervous system it now influences existed in its current form. Whatever relationship is running between parasite and host, in the ant colony, in the rat population, in the 40% of humans carrying Toxoplasma without knowing, it doesn't require the host's awareness to proceed. It only requires the chemistry to keep finding what it's looking for. And somewhere in the evolutionary record, across 48 million years of amber and inference and unrecoverable failure, something learned to look in exactly the right place. The movement looks normal, that's the first thing. An infected cricket walking toward a stream doesn't stumble. It doesn't twitch or drag or signal in any visible way that something internal has been rerouted. It walks the way a cricket walks, legs moving in their particular mechanical sequence, antennae reading the air, and if you didn't know what to look for, you would see nothing unusual at all, just a cricket, moving through the grass, doing what crickets do, except crickets avoid water. Except this one isn't. There's a specific strangeness to watching behaviour that looks chosen and knowing it isn't. Not because it proves something disturbing about free will, that's a different conversation, a heavier one, not for tonight, but because of what it reveals about how behaviour is built in the first place. What we recognise as movement, as decision, as an animal doing a thing is in most cases an output. The visible end of a long chain of chemistry. Change the chemistry quietly enough and the output changes. The animal doesn't change. The legs are the same legs. The sensory organs are reading the same world. Something upstream of all that, in the signalling, in the molecular instructions reaching the muscle and the nerve, has been adjusted. And the adjustment is invisible from the outside, which is part of why it took researchers a long time to find where exactly the instruction was being altered. In the case of Toxoplasma gondii and rodents, what changes is fear, specifically the fear of cat odour. A healthy rat encountering the smell of cat urine will move away from it. This is not a learned response. It doesn't require a rat to have ever seen a cat. The avoidance is encoded, heritable, produced by neural circuitry in the amygdala that responds to predator chemical signals the way a knee responds to a tap. automatically, reliably, without deliberation. In a rat carrying Toxoplasma, this response is muted, not erased entirely. The rat still processes the odour, still generates some neural response to it. But the downstream behaviour that should follow, the moving away, the elevated heart rate, the freeze and reassess, those either don't fully materialise or materialise weakly, or in some documented cases flip into something closer to attraction. The rat doesn't run, the cat catches the rat. Toxoplasma, which requires a feline gut to complete its reproductive cycle, gets where it needs to go. The body was borrowed just long enough. What stands out most to me, I think, is not the manipulation itself. It's how targeted it is. Toxoplasma doesn't suppress all fear in the rat. Anxiety around open spaces, around unfamiliar objects, around other potential threats, these remain largely intact. The alteration is narrow, specific, predator odour avoidance and not much else, which suggests that whatever the parasite is doing to the dopamine and testosterone systems in the amygdala, it has been refined across generations of rodents and parasite lineages to touch as little as possible while changing the one thing that matters. A blunt alteration would be detectable, correctable, less reliable. What evolved instead is surgical in a way that no surgery ever intended. There's a movie, you may have seen it, where an alien symbiote bonds with a human host and takes deep offence at being called a parasite. The character insists on something closer to partnership, mutual benefit. Two organisms, one body, both getting something from the arrangement. It's fiction, obviously, but the instinct behind the objection is interesting. Parasite carries a particular weight. It implies pure extraction, a relationship with no reciprocity, something taken without anything given back. And in most of the cases we're discussing tonight, that framing holds. The cricket doesn't benefit from walking into water. The ant doesn't benefit from the grip that kills it. But not always. The biology gets complicated at the edges. There are parasitic relationships that have drifted across time towards something harder to categorize, where the chemical presence of the organism has become load-bearing in the host's physiology in ways that weren't originally designed, where removing the guest produces its own disruption. Toxoplasma in humans, which we'll return to later, sits in that uncertain territory. Most carriers show no acute symptoms. Some data suggest subtle effects on personality and reaction time. Whether any of it constitutes benefit is genuinely unclear. The parasite isn't trying to help, but the body has, in some cases, quietly accommodated the presence, folded it into its baseline, continued around it. Venom would probably still object to the label. The biology, at least, would find the question more interesting than the answer. The ant climbing the plant stem is using its own muscles. This is worth repeating slowly because it changes something about how the whole thing feels. The fungus doesn't move the ant the way you might move a puppet. There are no direct connections, no mechanical override. What Ophiocordyceps produces are compounds, chemicals that enter the ant's body through the cuticle, migrate into muscle tissue and alter the calcium ion cycling that controls how muscle fibers contract. The neurons are still the ant's neurons. The signal travels through the ant's own nervous system. But somewhere between the signal and the muscle's response, something has been added to the conversation, and the conversation now ends differently than it would have without the addition. The ant climbs because its muscles are being told to climb, and its muscles are being told to climb because a fungus, several weeks into its own life cycle, has reached the stage where it needs to be elevated above the forest floor for its spores to disperse effectively. There is no cruelty in this. There is also no kindness. There is just chemistry, doing what it has been shaped to do. And you've probably felt at some point the slightly unnerving sensation of doing something automatic, reaching for your phone, taking a familiar route home, saying a word before you consciously decided to say it, and noticing a beat too late that you weren't exactly in charge of that moment. The neuroscience of that feeling is its own long conversation, but there's something in it that connects to this, gently. The line between chosen behavior and produced behavior is not always as clear as it feels from the inside. What the parasite exploits is a gap that already exists in how behavior is generated. Not a flaw, just the architecture. The chemistry that runs behavior is accessible because it has to be. Because hormones and signals and environmental inputs are supposed to alter it. That's what it's for. The parasite is, in a stripped-down sense, just another input, one the body never learned to screen for. The cricket reaches the stream. The water is cold, you can imagine it. The temperature dropping as the grass gives way to bank, the surface reflecting whatever light is left. The cricket enters. The hairworm that has been living inside it, coiled and patient, exits through the body wall into the water. The cricket sometimes survives this, not always, but sometimes it crawls back out, missing its passenger, and continues. Whether what continues is meaningfully the same cricket, same chemistry, same behavioral baseline, same animal it was before the worm arrived, is a question the biology doesn't answer cleanly. It moves. The legs still work. It finds its way back to the grass. A lock doesn't know what made the key. It only knows whether the shape fits, whether the tumblers move, whether the door opens or stays closed. The lock has no memory of being manufactured, no record of what the original key looked like, no way to distinguish a copy from the original if the copy is precise enough. Receptor sites in the body work something like this, and this is the one place tonight where the metaphor is almost too accurate to feel like a metaphor at all. Receptors are proteins, specific shapes embedded in cell membranes, waiting for molecules that fit them, neurotransmitters, hormones, signaling compounds produced by the body's own chemistry. When the right molecule arrives and binds, something happens downstream. A channel opens, a cascade begins, a cell changes what it's doing. The system evolved to respond to the body's own signals, but a signal's just a shape, and shapes can be mimicked. This is where the biology of parasitic manipulation becomes, to me at least, genuinely strange. Not in a disturbing way, more in the way that a very simple fact can suddenly feel enormous when you look at it from the right angle. Some parasites produce compounds that are structurally similar enough to the host's own neurotransmitters that the receptor can't reliably tell the difference. Not because the receptor is faulty, because the parasite's compound was shaped across enormous spans of evolutionary time to fit. The door opens. Whatever was supposed to happen when that receptor was activated happens, except the signal didn't come from the host's own chemistry. It came from something else entirely, something that has been refining its molecular shape across millions of generations of failed fits and occasional successes. The lock opened. The key just wasn't the original one. Spinochordides teleniaul, the hairworm that grows inside grasshoppers and crickets, produces proteins that structurally resemble the host's own neurotransmitters, specifically compounds that interact with the systems governing locomotion and the processing of light. The grasshopper's movement toward water isn't random, it's phototactic. The infected animal becomes drawn to light reflected off water surfaces. The worm doesn't push the grasshopper toward the stream. It adjusts what light means to the grasshopper's nervous system, at the receptor level, until moving toward reflected light becomes what the grasshopper does. The worm never touches a neuron directly. It just changes what the neuron hears. There's something worth pausing on here, even briefly. The proteins Spinochordides produces aren't crude approximations. Researchers who've analysed them found they share significant structural and functional similarity with the grasshopper's own nervous system proteins, a degree of molecular precision that doesn't happen by accident and didn't happen quickly. This is the result of a co-evolutionary process where every generation of worms that produced a slightly better fitting compound was slightly more likely to complete its life cycle. Millions of generations, tiny incremental adjustments, a molecular shape that grew slowly into something the receptor couldn't screen out. The receptor was never updated to exclude it. There was no selection pressure on the grasshopper to close that particular door, because closing it would have meant changing how the grasshopper's own nervous system functioned. The door couldn't be locked without locking out the original key too, so it stayed open. Toxoplasma works differently, but arrives at a similar outcome through a different molecular route. It doesn't mimic neurotransmitters directly. Instead, it carries a gene, a functional gene, encoding a working enzyme for the production of tyrosine hydroxylase. This enzyme is involved in the synthesis of dopamine. The parasite, once inside neural tissue, begins producing it. The host's dopamine levels shift. Locally in the tissue surrounding the cyst, dopamine is involved in reward processing, motivation, risk assessment. The change is subtle, but subtle in a system as finely tuned as mammalian reward circuitry is enough. The fear response that should accompany cat odor, which runs partly through dopamine pathways, doesn't fire with its usual intensity. The rat moves differently, not wildly, just slightly less cautiously than it should. What I find myself uncertain about, and I don't think the uncertainty is false modesty, is whether mimicry is even the right frame for all of this. Mimicry implies intent, a copy made to deceive, but the compound doesn't know it's mimicking anything. The gene encoding the enzyme doesn't know what dopamine is for. These are just shapes that fit, selected because they fit, retained because fitting them led to survival. The deception, if that's what it is entirely structural. A key that looks like no key you've ever seen opens a door that was never meant to be opened from the outside. And here the metaphor quietly runs out of room, because a lock and key is a human object, built with intent, operating between two parties who both exist. The receptor doesn't have a party on the other side, it just responds to what arrives. What arrives in these cases is something the receptor was never built to refuse. The cascade begins, the channel opens, something downstream changes in a direction that has nothing to do with the host's own survival and everything to do with the survival of whatever produced the compound. The body responds the way it always responds to that particular shape. It just never knew that shape could come from somewhere else. Fear has a texture. Not metaphorically, there is actual texture to it, at the level of what the body does when it arrives. The chest tightens in a specific way, different from exertion, different from cold. The gut shifts. Something behind the sternum changes quality briefly before the cognitive layer catches up and names what's happening. You've felt this. Most people feel it before they understand it, every time. The body moves first, the word comes after. This is not a flaw in the system. This is the system working exactly as it was built to work. The amygdala doesn't wait for interpretation. It receives sensory input, compares it against a library of associated threats built across your entire life, and, deeper than that, across evolutionary time, and it acts. The signal goes out before you've finished processing what you saw or smelled or heard. By the time you think that was startling, your heart has already accelerated, your pupils have already widened, your muscles have already received the instruction to be ready. The whole process from input to physical response takes milliseconds. In a rat, the equivalent process is older and in some ways simpler. Fewer cognitive layers between the sensory input and the behavioral response, a more direct line from smell to action. Cat urine contains compounds called felinine and trimethylamine. A rat that has never encountered a cat in its life will, upon smelling these compounds, initiate avoidance behavior. Move away from the source, increase alertness, reduce exploratory behavior in the area. This response is innate, encoded in the rat's neural architecture before the rat was born, shaped by predation pressure across so many rodent generations that it no longer requires any individual experience to activate. It arrives automatically. The smell comes in, the behavior follows, except in rats carrying Toxoplasma gondii. What the parasite does to this system is not dramatic. That's the part that stays with me, I think, more than anything else about this particular relationship. It doesn't flood the rat with false confidence. It doesn't override the fear system entirely, doesn't produce some pharmaceutical-grade suppression that leaves the rat functionally anesthetized to danger. What it does is quieter than that, and quieter in this context turns out to be much more effective. The amygdala still receives the signal. The felinine still arrives at the olfactory receptors, still gets processed, still generates a neural response that travels to the fear circuitry. The rat isn't rendered smell blind or neurologically incapacitated. What changes is what happens after the signal arrives. The downstream processing, the part where the fear signal normally amplifies itself, recruits dopamine pathways, generates the sustained behavioral avoidance that keeps the rat away from cat territory. That part doesn't fully run. The feeling that should arrive doesn't arrive with its usual weight, or it arrives briefly, shallowly, and then something about the chemistry in the tissue surrounding the Toxoplasma cysts dampens what comes next. The rat notices the smell. It just doesn't move away from it the way it should. Toxoplasma gondii forms cysts, small walled structures, in neural tissue, the brain. The amygdala specifically shows high cyst density in infected rodents, which is not random. The parasite doesn't distribute evenly throughout the brain. It accumulates in the region most directly involved in processing threat. Inside and around those cysts, the local chemistry changes. Toxoplasma carries functional genes for tyrosine hydroxylase, an enzyme involved in dopamine synthesis. The parasite settled into neural tissue begins producing it. Dopamine levels in the immediate area shift. And because the amygdala's fear response runs partly through dopamine pathways, that shift matters. There's also evidence of altered testosterone levels in infected male rats, higher than uninfected controls, which may contribute to reduced threat sensitivity through pathways still being mapped. The full picture is not entirely clear. Several mechanisms appear to be operating simultaneously, and researchers continue to argue, carefully and with good data on multiple sides, about exactly which one is primary. What isn't argued is the behavioral outcome. The rat moves differently, not erratically, just with less of the specific caution that should accompany the smell of a predator. Have been quietly turned down without its knowledge or consent. Moving through a world that contains a predator it no longer properly fears. I understand that instinct, but I think it's worth gently setting it aside for a moment, because it imports a framework of loss that doesn't quite fit the biology. The rat has no access to what it would have felt without the infection. There is no internal comparison happening, no ghost of the original fear response haunting the muted one. The rat is not experiencing diminishment. It is just experiencing its own chemistry, the only chemistry it has, the only baseline it knows, and that chemistry has been quietly adjusted in one narrow way. The absence of fear, when fear is absent, doesn't feel like anything at all. Toxoplasma gondii infects warm-blooded animals broadly, birds, pigs, sheep, humans, but it can only sexually reproduce inside the feline gut. Cats are the definitive host. Every other animal is, in the parasite's life cycle, a holding pattern, a place to wait. The behavioral manipulation in rodents is the exit strategy. Move the rat toward the cat, complete the cycle, reproduce. What makes this particularly striking biologically is the specificity. Infected rats don't show generalized fearlessness. They don't wander into open spaces they'd normally avoid, don't become broadly reckless in ways that would expose them to other threats. The reduced response is specific to cat odor, or at minimum most pronounced there, most reliably documented there, which implies a degree of precision in what's being adjusted. Not the whole fear system. The part of the fear system pointed at cats. Whether this precision is truly as targeted as it appears, or whether other fear responses are also subtly affected in ways that haven't been as carefully measured, is still an open question. I find myself inclined to think the picture is probably more complex than the clean version suggests, that what looks like surgical specificity might be a broader alteration that happens to be most visible in the cat odor response because that's where researchers looked first. But even the clean version taken at face value is strange enough. Somewhere between 30 and 50% of humans carry Toxoplasma gondii. Most acquired it through contact with cat feces, or through undercooked meat from intermediate hosts, or through soil. Most never developed acute illness. The immune system contained the infection, the parasite formed cysts, and life continued. The cysts persist. They are not cleared. They sit in neural tissue, in humans distributed more broadly than in rodents, less concentrated in the amygdala, and remain there for the life of the host, in most cases causing no clinical symptoms. Whether they do anything else is where the data becomes harder to read cleanly. Associations have been found in various studies between Toxoplasma seropositivity and reaction time, risk-taking behavior, certain personality measures, testosterone levels in men. The associations are real in the statistical sense. What they mean, whether Toxoplasma is causing these differences or correlating with conditions that make infection more likely, whether the effect size is biologically meaningful in any individual, these questions don't have settled answers. What can be said is this: the same parasite that quietly adjusts dopamine chemistry in rodent amygdalae is also present in the neural tissue of roughly a third of all humans alive right now. What it is or isn't doing there is not fully known. The feeling that should arrive and doesn't, that's not something you would notice. That's the part that takes a moment to absorb. The muted fear response in the rat produces no signal to the rat that something is missing. There is no sensation of absence. The chemistry simply runs its slightly altered course, and the behavior that follows reflects that alteration without announcing it. You can't feel a receptor that isn't firing at full sensitivity. You can only watch from outside as an animal moves towards something it should be moving away from, and piece together, slowly, across years of careful research, what the chemistry inside it must be doing to produce that specific quiet deviation from what fear is supposed to look like. Most of what the body contains it never announces. The bacteria outnumbering your own cells, the dormant viral sequences woven into your genome from infections your ancestors survived, the microscopic passengers moving through tissue that your immune system has assessed, contained, and, in a decision that looks almost like pragmatism, decided not to fully expel. The body is not as singular as it feels from the inside. Toxoplasma gondii was first identified in 1908 in the tissues of a North African rodent called a gundi. Two researchers working independently, Charles Nicole in Tunis, Adolphe Manceau in Paris, noticed the same crescent-shaped organism in their specimens and named it from the Greek toxin meaning bow or arc for the curve of its body. The year is worth holding for a second, 1908, less than 120 years ago, and yet parasites themselves had been noticed, documented, named, theorized about for thousands of years before that. Ancient Egyptian medical texts from 1550 BCE describe intestinal worms in enough detail to be recognizable. Greek physicians wrote about parasites they observed in patients and animals. Roman writers documented them. The presence of organisms living inside other organisms was not a modern discovery. It was a very old observation. What was missing, for most of that history, was any framework for understanding what a parasite actually was at the biological level. The ancients saw the effect. They named the creature. They had no way of knowing about the cell, the cyst, the receptor, the dopamine pathway. The knowledge sat at the surface for millennia, accurate in its observation and incomplete in everything below it. What Nicole and Manceau found in 1908 was the organism. What came after, slowly across the following decades, was the mechanism. Toxoplasma's full life cycle wasn't understood until the 1970s. Specifically, the discovery that cats were the definitive host, the only animal in which the parasite could complete sexual reproduction, came in 1970, when researchers in multiple laboratories identified oocysts in feline feces and traced the transmission route. Before that, the parasite's presence in warm-blooded animals was documented, but the logic of why it was there, what it was doing, where it was ultimately trying to go, that was missing. 62 years passed between the identification of the organism and the understanding of its life cycle. This is not unusual in parasitology. It's actually fairly typical. The behavioral manipulation component came even later. The first careful studies suggesting that Toxoplasma-infected rodents behaved differently around cat odor were published in the late 1990s. The dopamine mechanism, the tyrosine hydroxylase gene, the local chemistry shift in amygdala tissue, that work came in the 2000s. So the timeline runs like this. Ancient observers noticed parasites in bodies. Modern researchers identified Toxoplasma in 1908. The life cycle is mapped in 1970. The behavioral effect in rodents is documented in the late 1990s. The molecular mechanism begins to be understood in the 2000s. What the parasite was doing the entire time had nothing to do with when humans figured it out. The current estimate for global Toxoplasma prevalence in humans sits somewhere between 30 and 50%, depending on region, diet, and exposure to cats and soil. In some populations, parts of France, Brazil, Ethiopia, seroprevalence exceeds 60 or 70%. These are large numbers, quietly large. The majority of infections produce no acute illness in immunocompetent adults. The immune system mounts a response. The parasite retreats into cyst form, and the clinical episode, if there is one, passes. Many people never know an episode occurred. The cysts persist indefinitely in neural and muscle tissue, but in most healthy adults they sit there without producing symptoms that surface into conscious experience. What the cysts may or may not be doing subtly, at the level of receptor chemistry, dopamine baseline, low-level immune modulation, is where the research gets careful and the certainty gets thinner. The honest position, I think, is that we don't fully know. The associations between Toxoplasma seropositivity and various behavioral and psychological measures are real in the data. They are also small in effect size, complicated by confounders, and not yet explained by a mechanism as clearly established as the rodent dopamine pathway. Whether what happens in an infected rat amygdala has any meaningful analogue in a human brain, a larger, differently organized, more cognitively layered structure, is genuinely unresolved. What is resolved is the number, 30 to 50%. Roughly 2 to 4 billion people carrying a parasite whose behavioral effects in its intermediate hosts are among the most precisely studied examples of manipulation in all of biology. The guest arrived in most cases without an appointment, and in most cases the host never knew to check. The ant is still alive when the biting begins. This is the part that takes a moment. Not dead, not sedated in any way researchers have been able to confirm, alive with functioning nervous system, intact musculature, the same biology it had yesterday. And it climbs. 25 centimeters, sometimes more, up a plant stem in the understory of a tropical forest. It finds a leaf, a specific leaf, north-facing in many documented cases, positioned at a height between 25 and 30 centimeters above the forest floor, in a humidity range between 94 and 95%. Then it bites down on the leaf vein, and it does not let go. What happens in the jaw is not neural. This took researchers a long time to find, partly because the obvious assumption was that the fungus was acting on the brain, hijacking motor neurons, overriding the central nervous system, running the behavior from the top down the way most manipulations are imagined to work. When they looked at infected ant brains under microscopy, they found something unexpected. The brain tissue was largely intact. The fungus wasn't there, it was in the muscles. Specifically, the mandible muscles, dense fungal cells infiltrating the muscle fibers directly, altering calcium ion dynamics, changing how the fibers contracted and whether they could release. The grip wasn't commanded from the brain. The brain may have had nothing to do with it at all. The jaw locked because the muscle was made to lock. There's a long pause I keep coming back to when I think about this particular finding. Not because it's horrifying, I don't think it is exactly, but because it quietly dismantles something about how we assume control works in biology. We tend to think top-down. Brain sends signal, body follows. The brain is where behavior lives, where decisions are made, where anything complex originates. Ophiocordyceps found a different architecture entirely. It skipped the command center. It went straight to the hardware. Whether the ant experienced anything during this, whether there is any meaningful sense in which we can ask what it was like, is a question biology can't answer and probably shouldn't try. But the muscles locked, the fungus fruited, the spores dispersed, the amber specimen, 48 million years old. Carpenter ant, mandibles clamped around a leaf vein, Ophiocordyceps associated bite marks visible in the plant tissue, the same distinctive scarring pattern produced by infected ants today, the same semicircular marks made by the same mandible geometry locked in the same terminal grip. 48 million years ago, this was already the finished version. Whatever evolutionary path produced this, whatever intermediate forms existed, whatever partial manipulations preceded the full one, none of that is recoverable. The fossil doesn't show the origin, it shows the arrival at precision. The thing we're looking at in tropical forests today, this specific and strange and functional relationship, was already running in its current form before most modern mammal lineages existed. Before grass covered the continents, before the Amazon basin looked anything like it does now, the ant bit down, the leaf held, the fungus fruited, same as now. Carpenter ants that encounter infected nestmates will, in some circumstances, carry them out of the nest and deposit them at a distance. The behavior is not perfectly reliable. It doesn't always happen, the detection mechanism isn't fully mapped, but it occurs often enough to be documented as a colony-level response to infection risk. What the healthy ants are detecting is chemical. Infected ants produce a different cuticular hydrocarbon profile. The surface compounds that ants use to recognize nestmates assess health, communicate identity. The infected ant smells wrong, not dramatically, not in a way that produces immediate alarm, but differently enough that some nestmates register the change and act on it. The colony is, in this loose sense, doing something that functions like immune surveillance at the social level. Not foolproof, not complete, but present. The infected ant, once deposited outside the nest, doesn't return. Whether it can't or doesn't, the research doesn't cleanly distinguish. It climbs, it finds the right leaf. The height and humidity and compass orientation converge. The jaw closes. I want to be careful here about overstating the precision. The 25 to 30 centimetre height, the north-facing orientation, the narrow humidity range, these parameters come from careful fieldwork, primarily in Thai and Brazilian forests, across specific Ophiocordyceps ant pairings. Other pairings, other forest systems, other species combinations show variation. The precision that shows up in the literature is real, but it's real within specific ecological context, not as the universal law governing all zombie ant fungi everywhere. There are more than 200 described species of Ophiocordyceps that infect insects. They don't all produce identical behavior. Some manipulate beetles, some manipulate flies. The mechanisms vary, the outcomes vary. What they share is the basic logic: alter the host's behavior to position the fruiting body optimally for spore dispersal. But the specific chemistry, the specific muscles targeted, the specific behavioral output produced, these differ across the genus. The carpenter ant version is the most studied, the most photographed, the one that gets described as precise because it has been measured most carefully. Precision sometimes is just what thorough measurement looks like. The fungal cells inside the muscle fibers are not passive. They're metabolically active, producing compounds that interfere with the actin-myosin interaction that normally allows muscle fibers to extend and release after contraction. The specific compounds involved are still being characterized. This is genuinely recent biochemistry, some of it published within the last decade. What's established is the functional outcome. The fibers contract. They don't release. The mandibles close around the leaf vein, and the mechanical properties of the muscle are altered enough that the grip is maintained without ongoing neural input. The ant could, in theory, die at this point. The grip would hold anyway. The fruiting body emerges from the back of the ant's head, usually from the membranous region between the head and thorax. It grows upward over several days, eventually producing a structure called a stroma, a stalk topped with spore-producing tissue. The spores, when released, fall onto the forest floor below, where foraging ants walk. The cycle begins again. What the forest floor looks like in areas with high infection pressure is something researchers describe carefully. Ant graveyards, sometimes clusters of dead ants on leaf undersides at that characteristic height, each one with a small fungal stalk emerging from the same anatomical point. In some locations, the density is high enough that ant colonies have shifted their foraging routes to avoid zones of heavy Ophiocordyceps activity. The colony learn to walk around the place where the floor is dangerous, not quickly, not consciously, through the slower learning of which foragers came back and which ones didn't. The amber ant bit down 48 million years ago, and the leaf held and the fungus fruited, and the spores fell, and somewhere in the forest below, another ant walked through them. We don't know if that ant was detected by its nestmates. We don't know if the colony shifted its route. We know the relationship continued. We know it's still running. We know the jaw closes at the right height, in the right humidity, on a leaf vein that will hold the weight of what comes next. The muscle locks, the brain intact and largely untouched, is just above it. Before anything visible changes, something invisible already has. This is how most of what happens inside the body works. The surface event, the behavior, the observable outcome arrives last. Long after the chemistry has already shifted, long after the cellular negotiation has been running quietly in tissue you can't see or feel or access in any direct way. By the time the ant climbs, the fungal cells have been in the muscle for days. By the time the rat moves toward the smell it should avoid, the cysts in its amygdala have been producing tyrosine hydroxylase for weeks. The behavior is the last thing. Everything before it is invisible. To alter a host's behavior without killing it, this is the central biological problem that every manipulative parasite has to solve, and it is, when you look at it carefully, a genuinely difficult one. The host's immune system is not passive. It surveys tissue constantly, identifies foreign proteins, mounts inflammatory responses, recruits immune cells to sites of infection. A parasite that simply invaded neural tissue and began producing compounds indiscriminately would trigger responses that could clear the infection or damage the tissue so severely that the host became non-functional, or kill the host outright before the parasite's own reproductive goals were met. A dead host ends everything. A bedridden host too sick to move ends the transmission chain almost as completely. What evolved instead across the lineages of parasites that are still running today is something more careful than invasion, something closer to negotiation, not intentional, not aware, but functionally resembling a process of finding exactly how much can be altered without triggering the response that would undo the alteration. There is a specific state the body enters when it hasn't slept. Not immediately. The first few hours feel manageable, even normal. The nervous system compensating through adenosine pressure and cortisol and the particular alertness that comes from knowing you're tired and trying to work around it. But somewhere past the 18-hour mark, something in the cellular maintenance that normally runs during sleep begins to fall behind. Inflammatory markers rise. Cytokine profiles shift. The glymphatic system, which clears metabolic waste from neural tissue during slow-wave sleep, hasn't run its cycle. The tissue is carrying more than it should. The immune system, under sleep deprivation, becomes simultaneously overactive in some pathways and suppressed in others. This is not a stable configuration. The body is running its surveillance systems on a chemistry that is slightly off baseline, making decisions about what is foreign and what is self with instruments that haven't been recalibrated. This matters here because the cellular environment that Toxoplasma and other neural parasites inhabit is not static. It responds to the host's physiological state: sleep, stress, immune activation, inflammation, and the parasite's ability to remain in its contained cyst form depends partly on a particular immune balance being maintained. Severe immune disruption in either direction can reactivate latent Toxoplasma from cyst form into its actively replicating tachyzoite stage. Most of the time in healthy adults, this doesn't happen. The immune system holds the containment, but the margin is not infinite, and sleep is one of the variables that affects where that margin sits. Ophiocordyceps manages this problem, the problem of not killing the host too early, through what appears to be a remarkably controlled infiltration. The fungal cells enter the ant through the cuticle, the hard outer surface, and then spread through the body cavity and into soft tissue. But they don't immediately invade the brain. They don't immediately invade the muscles either. There appears to be a period of systemic presence, fungal cells distributed through the hemolymph, the ant's circulatory fluid, before the targeted infiltration of mandible muscle tissue begins. What governs the timing of this progression is not fully understood. The fungus appears to respond to host physiological signals, possibly the ant's own hormonal cycles, possibly chemical signals from the muscle tissue itself, in a way that staggers the invasion so the host remains functional, mobile and capable of the climbing behavior the fungus requires until the moment it no longer needs the host to be any of those things. The host stays alive because being alive is useful, until it isn't. Toxoplasma's containment strategy is different, but runs on a similar logic. The acute phase of infection, the tachyzoite stage, where the parasite replicates rapidly, is the dangerous phase, the one the immune system mounts its strongest response against. If the acute phase continues too long, the inflammation it produces could damage neural tissue in ways that compromise the host's survival. What Toxoplasma does under immune pressure is convert. The rapidly replicating tachyzoites transform into bradyzoites, slow replicating forms enclosed in thick-walled cysts. The cyst wall is resistant to most immune attack. The bradyzoites inside replicate slowly enough that the metabolic activity is low, the foreign protein production is reduced, the inflammatory signal is dampened. The immune system detects the cysts, surveys them, and in immunocompetent hosts largely leaves them in place, unable to fully clear them, but able to prevent them from reactivating. This is the chronic phase, the one that lasts for years or decades or the remainder of the host's life. The parasite is contained. The host is functional. The cysts sit in the tissue, waiting for the transmission event, the predation in rodents, that will complete the cycle. Both parties continue. Neither exactly wins. What I find worth sitting with somewhere in all of this is how much the manipulation depends on restraint. Not the restraint of a conscious actor making a decision, but the functional restraint that emerges when anything that goes too far gets selected against. The fungal lineages that killed their host too quickly didn't fruit. The Toxoplasma strains that triggered too much inflammation got cleared. The hairworm chemicals that disrupted too many neural systems produced hosts that couldn't reach water. The ones that are still running, the relationships we can study, are the ones that found, across enormous spans of trial and elimination, the minimum effective alteration, enough to redirect the host. Not so much that the host stops being useful. It is, in a stripped-down sense, the biology of just enough. Right now, somewhere in the cells of roughly a third of all people alive, bradyzoites are sitting in cyst walls, metabolically slow, chemically quiet, waiting in tissue that has folded around their presence and continued. The host is asleep in most cases, at some point in the next several hours. The glymphatic system runs. Inflammatory markers return toward baseline. The immune surveillance that monitors the cysts recalibrates. The containment holds. The chemistry that has been running beneath sensation, beneath awareness, beneath anything that surfaces into conscious experience, continues in the dark. Most of what the body manages, it manages without telling you. You don't feel the negotiation, you only feel the quiet that follows when it holds. It begins as something too small to see, an egg deposited in fresh water hatching into a larva called a coracidium, free-swimming, microscopic, carrying inside it the full developmental potential of an organism that will eventually be longer than the animal it lives inside. The larva doesn't know this. There is no knowing involved. There is only the water and the movement and whatever comes next. What comes next is ingestion. A mayfly larva, or a water-dwelling insect, swallows the coracidium. Inside that host, the hairworm insists, pauses, waits, wraps itself in a protective layer, and sits in the tissue, metabolically patient for days or weeks. Then a cricket eats the mayfly. Inside the cricket, the cyst dissolves. The hairworm emerges into the body cavity, the hemocoel, the open circulatory space, and begins to feed. Not on organs directly, but on the nutrients circulating in the hemolymph. It absorbs, it grows. This is the part that takes a moment to absorb proportionally. Spinochordodes tellini, fully grown inside its host, can reach a length of 30 to 100 centimeters. The cricket it inhabits is typically 2 to 3 centimeters long. The worm coils inside the body cavity in dense loops, occupying space that the cricket's own organs have been gradually compressed to accommodate. The digestive organs, the reproductive tissue, the fat body that would normally fuel the cricket through winter. The cricket during this period eats more than usual, forages longer. The hairworm is consuming resources, and the host's body is, in some documented cases, compensating by increasing feeding behavior, a change that may itself be chemically induced, though the mechanism is less studied than the terminal water seeking that follows. Earlier in this video, back when we were watching a cricket walk toward water with the mild, uncanny feeling that something was off, I asked half seriously whether you'd like to try calling venom a parasite to his face. The hairworm might actually give venom pause, not because it's violent or dramatic, or it isn't from the outside, but because what it does to the definition of where one organism ends and another begins is genuinely difficult to resolve. For the weeks or months that Spinochordodes tellini is growing inside the cricket is eating for it, moving for it, keeping it warm, keeping it oxygenated. The worm is, in a functional sense, running significant portions of the host's metabolic life. Parasite feels accurate, but so uncomfortably does something else. The water-seeking behavior begins when the worm is ready to exit. The cricket, which under normal circumstances avoids open water with the reliable instinct of an animal that cannot swim well and knows it, begins moving toward it. The behavior is not frantic. It doesn't look desperate or disordered. The cricket walks, normally at its usual pace, in the direction of water. The compounds responsible are proteins produced by the worm that interact with the cricket's own neurotransmitter systems, specifically proteins structurally similar to the cricket's WNT signaling molecules, a family of proteins involved in neural development and, in adults, in the regulation of certain behaviors. The worm's proteins don't replace the cricket's. They add to the conversation, changing what the signaling says about light, about direction, about what the cricket's body understands as the right way to move. The cricket reaches water, enters it. The exit is fast. The worm emerges through the body wall, or sometimes through the cricket's mouth or anus, within minutes of the host entering water. It uncoils into the water, free for the first time since it was a larva, and begins its adult life. The adult hairworm doesn't feed. It has no functional digestive system. Its only biological purpose at this stage is to find another hairworm, mate, and deposit eggs. Has days, maybe weeks, to do this. The cricket, as noted in an earlier chapter, sometimes survives, crawls back onto land, body wall intact or partially healed, missing the dense coiled weight it carried for months. Whether the cricket's behavior normalizes after, whether the WNT signaling proteins dissipate, whether the water-seeking stops, hasn't been studied in enough surviving individuals to say cleanly. The egg that started this was too small to see. What it became was longer than the animal that carried it, coiled in loops in a space that kept shrinking, patient in a way that has nothing to do with patience, waiting for the chemistry to reach the stage where water was the only direction that made sense. The cricket walked toward it. The water was there. The immune system is not a single thing. This sounds obvious when stated plainly, but it's worth holding for a moment, because most of the ways we talk about immunity treat it as though it were a unified defense, a wall, something either up or down, active or inactive. The reality is considerably more distributed than that. There are innate responses and adaptive responses, humoral pathways and cellular ones, regulatory mechanisms layered over inflammatory ones, feedback loops that dampen what other feedback loops amplify. The whole architecture is less a wall and more a conversation, ongoing, adjustable, full of competing signals that have to be continuously balanced against each other. What parasites that survive long-term inside a host have learned to do across evolutionary time is not breach the wall. They've learned to enter the conversation. Inflammation is the immune system's loudest response. When tissue is damaged or foreign material is detected, inflammatory signaling begins. Cytokines are released, blood flow increases to the area, immune cells are recruited, the local environment becomes hostile to whatever triggered the response. Done correctly, inflammation clears infections, heals wounds, removes debris. It is essential. It is also metabolically expensive, tissue-damaging if sustained, and in some circumstances more dangerous to the host than whatever triggered it. The body knows this. Regulatory mechanisms exist specifically to constrain inflammation, to turn it on when needed and off when the job is done, to prevent it from running past the point of usefulness into the territory where it begins damaging healthy tissue. Parasites that have been living inside hosts for millions of years have, in several independent lineages, found ways to engage these regulatory mechanisms. Not to suppress immunity entirely, a host with no immune function is a host that dies quickly of secondary infections, which ends the parasite situation just as completely. What they manipulate specifically is the balance between inflammatory and anti-inflammatory signaling, nudging it toward the quieter end just enough to make their own presence tolerable. Toxoplasma gondii, in its chronic cyst form, actively modulates the immune environment surrounding it. The cyst wall itself is part of this. It's not just a physical barrier, but a biochemically active surface that reduces the inflammatory signal the cyst generates. The bradyzoites inside produce proteins that interfere with cytokine signaling in the surrounding tissue, specifically dampening the production of interleukin-12 and interferon gamma, two of the primary drivers of the anti-toxoplasma immune response. This is precise in a way worth pausing on. IL-12 and IFN-gamma are exactly the cytokines that, if expressed at full intensity around the cyst, would trigger the immune response most likely to clear it. The parasite doesn't suppress all cytokines. That would compromise the host's ability to fight other infections. It suppresses, with some specificity, the ones most dangerous to itself. The host's immune system continues surveilling the cyst. It detects it. It responds to it, partially. But the response is muted just enough that the cyst remains viable, contained but not cleared, generation after generation of bradyzoites living in a space that the immune system has, in effect, decided to maintain rather than destroy. There's a word for this kind of arrangement in immunology: tolerance. The immune system develops tolerance to self-tissue during development, learning to distinguish the body's own cells from foreign material. In some parasitic infections, what develops over time is something that functions like acquired tolerance. The immune response to the parasite becomes calibrated, restrained, persistent without being eradicating. The parasite becomes, in a narrow biological sense, part of the host's immunological landscape. I find this genuinely harder to categorize than straightforward infection. The language of invasion doesn't quite capture a relationship that has been running for decades inside a host whose immune system has incorporated the parasite's presence into its baseline calculations. It's not acceptance. It's not partnership. It's something in between that biology hasn't settled on a clean word for. The broader picture across parasitic lineages shows similar strategies arrived at independently. Filarial nematodes, the worms responsible for lymphatic filariasis and river blindness, produce compounds that expand regulatory T-cell populations in the host, shifting the immune environment towards tolerance. Schistosome flatworms alter the balance of Th1 and Th2 immune responses, reducing the inflammatory Th1 activity that would target them most effectively. Leishmania parasites survive inside macrophages, the immune cells specifically designed to destroy pathogens, by inhibiting the very killing mechanisms those cells use. Each mechanism is different. The logic is the same. Find the regulatory lever that controls the response most dangerous to your survival. Apply enough pressure on it to reduce that response below the threshold where it becomes eradicating. Leave everything else running. The host continues. The infection continues. The conversation in the immune system continues, slightly rewritten, in ways the host cannot feel. You already know what it feels like when inflammation is running. The heat of a healing cut, the swelling around a splinter, the full body ache of a fever working against an infection. These are not subtle experiences. The immune system at full inflammatory output announces itself clearly. What parasitic immune modulation produces is the opposite of announcement. It produces nothing the host can feel. No sensation of something being suppressed. No awareness of a regulatory mechanism being engaged. No signal that the immune conversation happening in the tissue surrounding a cyst has been quietly edited. The host goes about its life, sleeping, eating, moving, while the balance of cytokine signaling in neural tissue is maintained at a specific calibration that keeps a foreign organism alive inside it. The quiet is not emptiness. It's the result of something working very hard to stay unnoticed. The difference between those two things, genuine absence and engineered silence, is invisible from the inside, which is, of course, exactly the point. There's a point, writing something like this, where the mind starts to lag behind the material. Not dramatically, just a slight softening at the edges. Sentences that take a beat longer to form. The particular flatness that settles in when concentration has been running for too long without rest. I've been with this script for hours now, and I notice it. The way the words come slightly slower, the way I have to reach a little further for the next thought. It feels relevant, actually, to this chapter specifically, because what we've been tracing across these pages is a picture of manipulation that can start to feel almost frictionless. The fungus that positions its host with geometric precision, the protozoan that finds the exact receptor, the worm that rewrites what water means to a cricket's nervous system. All of it clean, all of it working. But it doesn't always work. Failure is built into all of this, not as an exception, as a structural feature of how these relationships operate. The behavioral manipulation is probabilistic, not guaranteed, and the difference between those two things matters more than the clean case studies suggest. An infected ant doesn't always reach the correct height. Some bite down too low or too high, or on the wrong surface, or not at all. The fungal spores that fall from a correctly positioned fruiting body land on a forest floor that may or may not contain foraging ants at that moment. The ants that pass through may or may not acquire an infectious dose. The ones that do may be detected by nestmates and removed before the manipulation completes. At every stage the chain can break. Toxoplasma in a rat produces muted predator avoidance, not absent predator avoidance. The rat is less cautious, not reckless. Some infected rats still avoid cat odor zones, less consistently, less reliably, but enough that transmission via predation is not certain. Some infected rats are never eaten. They live out their lives with cysts in their amygdala, and the dopamine shift running quietly in the life cycle never completing. The parasite in those individuals ends. No catgut, no sexual reproduction, no oocysts, just a cyst in aging neural tissue in an animal that eventually dies of something unrelated, and the bradyzoites inside it die too. This happens constantly, in most infections probably. I keep thinking about this, Maybe because my own thinking is slower right now, running on less than it should. But there is something in the failure rate that reframes the whole picture. We study the successes. The amber fossil is a success. The documented cases of water-seeking crickets are successes. The predation studies in rats showing elevated Toxoplasma transmission are successes. The literature is built from cases where the manipulation worked well enough to observe and measure and publish. What's invisible is the background rate of incomplete manipulations, partial effects, broken chains. The ant that climbed to 22 centimeters instead of 27. The rat that avoided the cat anyway. The grasshopper that reached the stream bank and turned back. These don't leave records. The manipulation's apparent precision is partly real and partly an artifact of what gets counted. There's also the immune system. Some hosts clear infections that other individuals of the same species cannot. Genetic variation in immune response genes means that a Toxoplasma infection producing stable chronic cysts in one rat may produce a more aggressive inflammatory response in another, one that damages the parasite's cysts, reduces bradyzoite viability, or in some cases approaches clearance. Not complete clearance in most documented cases, but suppression significant enough that the behavioral effect is reduced or absent. The manipulation requires the cyst to survive. The cyst requires a particular immune balance to survive. That balance varies between individuals. Some individuals never tip into the chronic phase that enables behavioral alteration. The infection runs its acute course, and something about the host's particular immune architecture prevents the stable cyst establishment that everything downstream depends on. No cyst, no dopamine shift, no behavioral change. The life cycle ends there. The hairworm fails too. Spinochordodes teleniae in a cricket that never encounters water, or encounters it in a location too small for the worm to survive after exit, or is eaten by a predator before the water-seeking behavior activates. In all these cases, the worm's life cycle doesn't complete. The adult stage, which needs open fresh water to find a mate, never arrives. The eggs are never laid. The chorocidea never hatch. A worm that spent months growing inside a cricket, coiling into every available space, consuming resources, reaching lengths that strain the definition of what can fit inside a 3-centimeter body. That worm sometimes simply ends in the wrong place at the wrong time, without completing a single step of the reproductive sequence it was shaped across millions of years to complete. There's no drama in this. That's something I keep coming back to in the state I'm in right now. Too tired to reach for significance, just looking at the thing plainly. The failure isn't tragic. The success isn't triumphant. These are just biological outcomes, distributed across populations, averaged across time, producing the net result that certain lineages continue and others don't. The ones that continue aren't better or more determined. They're the ones that across enough attempts had enough successes to persist. Everything we've looked at tonight is a survivor of its own failure rate, including, in a loose sense, most of what's alive. There's something I want to tell you about coffee before we get into the genetics. Not a detour, it connects, I promise, in the way that the best biological connections do. Sideways through this something you wouldn't expect. But first, coffee. Specifically what caffeine actually does, which is not what most people think it does. Caffeine doesn't give you energy. It borrows it. What it does, precisely, is block adenosine receptors in the brain. Adenosine is a compound that accumulates in neural tissue across the hours you're awake, a chemical record of wakefulness, building slowly, binding to receptors that produce drowsiness when activated. The longer you're awake, the more adenosine, the stronger the signal to sleep. Caffeine fits the same receptors without activating them. It sits there, blocking the adenosine from binding, and the drowsiness signal doesn't fully arrive. The tiredness is still accumulating. The adenosine is still being produced. You just can't feel it for a while. I mention this because I am, at the time of writing this chapter, considering a very large cup of coffee. The adenosine in my own neural tissue has been building for longer than it should have. The receptors are waiting. The signal is there. I just keep blocking the feeling of it, which is, when you put it next to everything we've been discussing tonight, a mildly funny thing to be doing voluntarily. The rat doesn't choose to have its fear response muted. I am more or less choosing to have my sleep signal muted. The mechanism is different. The basic structure, a receptor that should be receiving one signal, receiving something else instead, is not entirely dissimilar. Venom would probably not appreciate being compared to caffeine, but here we are. Now, the genetics. Some prey species show fear responses to predator chemical signals they have never personally encountered. A rat raised in a laboratory, no cats in its entire life, no cat odour in its environment, will still initiate avoidance behaviour when felinine is introduced to its cage. The response is there before the experience. It was there at birth. This is not unusual in biology. Innate behaviours, encoded responses that don't require learning to activate, exist across enormous ranges of taxa. What makes predator odour avoidance interesting is how specific it is. Not just fear of novel smells, not generalised neophobia, but a targeted response to particular compounds produced by particular predators. The rat's genome contains, in some functional sense, a record of the cat, not a memory, a preparation. The mechanism runs through the olfactory system into the amygdala, through specific receptor pathways that are genetically determined. The receptor proteins that detect felinine are encoded. The neural connections that carry that signal to fear circuitry are laid down during development. The behavioural response that follows, avoidance, freezing, elevated stress hormones, is the output of a system assembled before the rat ever took a breath. What this means is that the wariness is structural, woven into the architecture of the animal before experience has any chance to contribute. And what Toxoplasma has to overcome in producing its behavioural alteration is not a learned caution that could theoretically be unlearned. It's something older, something laid down in the genome across generations of rodents that were eaten by cats, the survivors being differentially the ones whose fear circuitry was most reliably activated by cat odour. The wariness survived because the rats without it didn't. This is the part I find myself returning to, even tired, even at this point in a very long script. What the parasite is bypassing is not a habit, not a preference, not a behavioural tendency that developed over an individual lifetime and could, in principle, be redirected by experience. It's bypassing the distilled outcome of predation pressure across an evolutionary timescale, a response so deeply encoded that no individual rat experience can produce it or remove it, only the slower process of which genotypes survive and which don't. The felinine response is in the rat because the rats without it were eaten, consistently, across enough generations that the encoding became fixed, and then something found a way around it anyway. There are other examples of innate predator recognition across vertebrates. Ground squirrels show alarm responses to the scent of California king snakes, a predator, but reduced responses to gopher snake scent, a non-predator. The distinction is encoded, not learned. Zebrafish larvae respond to chemical alarm cues released by injured conspecifics with innate fear behaviour before their nervous systems are fully developed. Domestic chicks show distress responses to overhead hawk silhouettes on their first exposure, a response that centuries of domestication haven't fully removed. The animal doesn't need to have been there before. The genome was there, in a sense, for all of it. What's interesting about parasite manipulation in this context is what it implies about the stability of innate responses. Innate behaviors are often described as fixed, hardwired, resistant to modification, the reliable baseline beneath all the learning and plasticity that sits on top. And in many contexts this is true. Learned behaviors can be extinguished. Innate ones are more stubborn. But Toxoplasma modifies the expression of an innate response. Not by altering the genome, not by removing the encoding. The rat still has the felinine receptor, still has the neural pathway, still has the amygdala circuitry that was built to produce avoidance. What changes is the downstream chemistry that determines whether the signal produces its full behavioral output. The encoding is intact, the execution is muted. It's the difference between a program that still exists on a computer and a program that runs at reduced capacity because something in the operating environment has changed. The instructions are all still there. Something is just intercepting the output. The adenosine is still accumulating. Yours probably right now, if you've been awake long enough, if the night has gone long, if the weight behind your eyes is the slow pressure of a signal that has been building for hours and is still waiting patiently for the receptor to open and let it through. The caffeine eventually clears, the receptors open. The signal arrives all at once, usually, which is why the crash after coffee feels like more than just returning to baseline. You're receiving everything that was held at the door while the blockade held. The rat's fear response, muted by parasite chemistry, sometimes partially returns when an infection is cleared or the cystload is reduced. Not always, not completely. But the encoding is still there, waiting in the architecture of the amygdala for the conditions that would let it run at full intensity again. Some signals just need the door to open. I should mention before we get into the snail that I'm currently experiencing what I can only describe as a full-body conspiracy. No sleep last night, hours of writing about organisms that hijack nervous systems, mute pheromosomal responses, and redirect behavior without the host's awareness. And now, sitting here, I am genuinely, irrationally aware of every slight itch, every small fatigue, every moment of unfocused attention, and some part of my very tired brain keeps suggesting unhelpfully that perhaps every parasite we have discussed tonight has somehow found its way into my particular body simultaneously. The Toxoplasma is muting my threat assessment. The hairworm is redirecting me toward the nearest body of water. The fungus is, presumably, measuring the humidity. This is not what is happening. What is happening is that I need sleep. The adenosine is winning. Anyway, the snail, Succinea putris, is a small amber-coloured land snail. It lives in damp vegetation near water, slow-moving, shade-seeking, spending most of its time in the lower parts of plants where light is filtered and humidity is high. It is not, under normal circumstances, a conspicuous animal. It moves the way snails move, which is to say slowly and with complete indifference to urgency, and it stays where the conditions suit it. Then Leucochloridium paradoxum arrives. The parasite enters through snail feces contaminated with bird droppings containing Leucochloridium eggs. The snail ingests them while grazing. Inside the snail's digestive system, the eggs hatch into miracidia, which penetrate the gut wall and begin developing into sporocysts, branching tubular structures that spread through the snail's body cavity and eventually migrate into the eye stalks. The eye stalks of a snail are thin, semi-transparent tubes. The eyes sit at the tips. Under normal circumstances, the eye stalks are unremarkable. Small, soft, retractable, the colour of the snail itself. When Leucochloridium sporocysts are inside them, they pulse. The sporocysts are banded, green and brown in alternating segments, sometimes with hints of yellow, and they contract rhythmically, producing a throbbing motion visible through the thin eye stalk wall. The effect from the outside is of something moving inside the snail's head, something striped and rhythmic, catching light, expanding and contracting at roughly the rate of a caterpillar's movement. The resemblance to a caterpillar is not coincidental. It has been shaped, across an evolutionary process that requires no designer and no intent, into exactly the visual signal most likely to attract the attention of insectivorous birds. The colour, the banding, the rhythm of the pulse, all of it converges on the appearance of a prey item that birds recognise and eat. The snail's behaviour changes. Succinea putris, infected with Leucochloridium, moves toward light rather than away from it, climbs higher on vegetation instead of staying in the lower, darker zones where it would normally remain, exposes itself to open areas during daylight hours, the eye stalks with their pulsing contents raised and visible, in exactly the locations where foraging birds are most likely to be scanning for movement. The snail doesn't retract its eye stalks the way healthy snails do when approached. The retraction reflex, a protective behavior present in uninfected individuals, is partially suppressed in infected ones. The eye stalks stay extended, the pulse continues, the bird sees movement. What the bird eats usually is just the eye stalk, not the whole snail. Birds peck at the pulsing structure, remove it, and the snail survives. Eye stalk gone, or partially gone, the Leucochloridium sporocysts now inside the bird's digestive system, where they can complete their sexual reproduction and release eggs in the bird's feces. The snail missing its eye stalk eventually regrows it. This takes weeks. In some documented cases, a new sporocyst migrates into the regrown eye stalk and the process repeats. The same snail, a new eye stalk, the pulse beginning again. There is something about this particular relationship that sits differently than the others we've covered. Partly the visual, the image of the pulsing eye stalk is specific enough that once you've seen it, it doesn't leave easily, but also something about the scale of the manipulation. The snail is not directed toward water. It's not directed toward a predator's scent zone. It's transformed visually into a lure. The parasite hasn't just redirected the snail's behaviour, it has made the snail's body into an advertisement. The snail is still there, underneath all of it. Still moving at snail pace, still processing whatever sensory information reaches it, still doing what snails do. The manipulation is running on top of that, changing what the body displays to the outside world, while the animal inside continues its ordinary existence. The bird flies away with the eye stalk. The snail sits on an exposed leaf in full daylight, which is not where it would choose to be. The regrowth begins, cellular repair, tissue regeneration, the slow biological process of rebuilding a structure that was removed. The immune system manages the wound. The remaining sporocysts in the body cavity wait. In a few weeks there will be something to migrate into again. The snail will climb, the eye stalk will pulse, the bird will be somewhere nearby or it won't, and the difference between those two outcomes is just probability. The same probability that governs everything else we've looked at tonight. The same background rate of success and failure and incomplete chains that makes this biology less like a mechanism and more like a very old, very patient kind of arithmetic. Most of the time something works, not always the same thing, not always for the same organism, but something somewhere in the forest, in the damp grass near the water, on the underside of a leaf at the right height in the right humidity, is completing a cycle that has been running since before most of the trees around it existed. Most stories about infection end at clearance. The pathogen is gone, the immune system won, the body returns to baseline and life continues from where it left off minus the disruption. This is the narrative arc that medicine is organized around: illness, intervention, resolution. And it's a useful arc because it's often accurate enough to act on. But accurate enough is doing quiet work in that sentence. What happens to a nervous system that has been chemically altered for weeks or months after the source of the alteration is removed is not always a clean return. Receptor systems that spent an extended period operating under modified conditions don't necessarily recalibrate instantly to their pre-infection baseline. Synaptic connections that were used differently during the infection period have been, at the molecular level, slightly remodeled by that use. The brain is not a static structure that returns to a stored configuration when a perturbation ends. It's a dynamic system that has been running continuously in a slightly different chemical environment, and it was changed by that, incrementally, in ways that may or may not fully reverse. The evidence for this in Toxoplasma is careful and incomplete, which is an honest place for it to be. Studies examining rodents after experimental clearance of Toxoplasma infection, achieved through antiparasitic treatment in laboratory settings, have found that some behavioral changes associated with infection persist after the parasite load is reduced. The felinin avoidance response, which is muted in infected animals, doesn't always fully return to uninfected levels immediately upon clearance. In some individuals it does, in others the restoration is partial or slow, or not clearly distinguishable from the infected state within the time frame of the study. What this suggests, carefully, without overstating, is that the dopamine pathway alterations produced by chronic Toxoplasma cysts may leave something behind. Not the parasite, not the active manipulation, but a residue in the receptor density, the baseline dopamine turnover, the sensitivity of the amygdala circuitry that was operating in modified conditions for an extended period. The guest has left. The room is slightly different than it was before the guest arrived. This is not unique to parasitic infection. It's actually a feature of how nervous systems work more generally. Extended exposure to any chemical that alters neurotransmitter dynamics tends to produce compensatory changes in the receptor systems involved. Downregulation of receptors when a signal is chronically elevated, upregulation when it's chronically suppressed. These compensatory changes are the nervous system attempting to maintain equilibrium in the face of persistent perturbation. They are adaptive while the perturbation is present. When the perturbation ends, the compensation may overshoot or normalize slowly, or in some cases settle at a new equilibrium that differs from the original. This is the basic biology behind tolerance and withdrawal in pharmacological contexts. The same principle applies more quietly to anything that spends months altering dopamine levels in amygdala tissue. The parasite and the pharmaceutical are doing very different things for very different reasons, but the nervous system's response to chronic chemical alteration doesn't particularly distinguish between them. In the Ophiocordyceps relationship, the question of what remains after the parasite is gone doesn't arise in the same way, because the host doesn't survive the relationship in most cases. The ant that bites down doesn't recover. There is no after for the host to occupy. But there are ants that are exposed to the fungus and don't complete the behavioral sequence. Individuals that acquire a sublethal fungal load, that are groomed by nestmates before the mandible muscle infiltration reaches completion, that are removed from the colony and deposited outside before the climbing begins. In these individuals, what the partial infection has done to muscle tissue, to hemolymph chemistry, to whatever neural environment was beginning to be altered, this is largely unstudied. The partial case is harder to find and harder to measure than the complete one. What happens in the middle of a manipulation that doesn't finish is a gap in the literature that is almost certainly full of interesting biology. There is something I keep returning to in this, and I want to be clear that it's tentative, more of a noticing than a conclusion. The framework we use for parasitic manipulation tends to focus on the active phase, the infected rat behaving differently, the ant climbing to the correct height, the cricket entering the water. These are the observable events, the ones that can be measured and published and cited. The before and after, the uninfected baseline and the post-infection residue, are harder to access and receive less attention as a result. But if chronic Toxoplasma infection produces lasting changes in dopamine receptor sensitivity in rodents, and if roughly a third of humans are carrying chronic Toxoplasma infections, and if the same basic neurotransmitter systems are involved in both, then the question of what the post-infection nervous system looks like in humans who have cleared an acute infection while retaining cysts is not a trivial one. It doesn't have a settled answer. The human data is correlational, the effect sizes are small, the confounders are numerous, but the question is there, sitting in the literature, not quite resolved, not quite dismissed. The room is slightly different than it was before the guest arrived. In most cases, slightly is the right word. Small shifts in receptor density, marginal changes in baseline chemistry, differences that fall within the range of normal variation and would be invisible in any individual without a controlled comparison. The kind of difference that only becomes visible when you average it across hundreds of individuals in a carefully designed study. Not dramatic, not debilitating, just the quiet biological record of something that was present for a long time and then, in the acute sense, wasn't. The nervous system adapted to it, then it adapted to its absence. Whether those two adaptations fully cancel each other out, whether the system returns exactly to where it began or settles somewhere fractionally different, is a question the current evidence can't cleanly answer. The ant's mandible muscles, in the individuals that survive partial infection, presumably continue working. The grip that was beginning to be altered, the calcium-ion dynamics, the actin-myosin interaction, the slow fungal remodeling of muscle fiber behavior. If interrupted early enough, the muscle may recover full function. The tissue repairs itself. The fibers that were being infiltrated are replaced through normal cellular turnover. But cellular turnover takes time, and in the days or weeks between partial infection and full recovery, the ant's grip is what it is, somewhere between its original state and what it was being made into. Most things recover. Most things recover slightly changed. The difference between those two statements is small, and it is also, in the long view of what nervous systems are and how they work, everything. Start with something simple. A rat moves away from a smell. This is behavior. The rat detected a chemical signal. The signal was processed. The processing produced movement in a particular direction. We watch it and we say, the rat did that. The behavior belongs to the rat. It originated in the rat's nervous system and was executed by the rat's muscles, and the rat is, in whatever sense we use the word, the author of it. This is how behavior is usually framed. A property of the organism, something the organism does. Now change one variable. The rat is carrying Toxoplasma. The dopamine chemistry in its amygdala has been running at a modified level for weeks. The fear response that should follow the predator odor signal is dampened and not absent dampened by a shift in receptor sensitivity produced by a protozoan that has been living in neural tissue and altering local cytokine and dopamine signaling for its own survival purposes. The rat doesn't move away from the smell, or moves away less decisively, or, in some documented cases, investigates it. Same rat, same muscles, same neurons, same olfactory receptors detecting the same compound, different behavior. Who authored that? This is not a philosophical question dressed in biological clothing. It's a biological question that happens to have philosophical weight, which is a different thing. The reason it matters biologically is that behavior is the unit we use to understand an organism's relationship to its environment, what it needs, what it avoids, what it seeks, how it has been shaped by the selection to respond to the world it lives in. If behavior can be systematically altered by the presence of another organism, then what we're observing when we observe behavior is not always a clean signal from the organism itself. It's sometimes a composite, the organism plus whatever is altering its chemistry. This isn't entirely new as a biological concept. Hormones alter behavior: testosterone, cortisol, oxytocin, a dozen others, and hormones are themselves produced in response to environmental inputs, social conditions, developmental history. The boundary between the organism and its chemical environment was never perfectly clean. Behavior has always been a product of the organism interacting with conditions inside and outside it. What parasitic manipulation adds is an agent, not an environmental condition, not a diffuse chemical gradient. A living thing with its own evolutionary history that has been specifically shaped across millions of years to produce a particular alteration in this particular host's chemistry for its own reproductive purposes. The hormone doesn't have a life cycle, the parasite does. That distinction changes something about how to think about the behavior that follows. I find myself genuinely uncertain about where to draw the line here, and I think the uncertainty is appropriate rather than a failure to think it through carefully enough. There is a version of this argument that goes quite far, that because behavior is always produced by chemistry, and chemistry can always be influenced by external agents, the concept of behavioral ownership becomes fuzzy across the board. That the rat was never purely the author of its own behavior in a strong sense. I don't think this version is very useful. It's technically defensible, but it dissolves a distinction that has real explanatory value. The difference between a rat behaving according to its own evolved chemistry and a rat behaving according to chemistry that has been specifically modified by another organism's evolved manipulation strategy is a meaningful difference, even if both are ultimately just chemistry. The question isn't whether the rat's behavior is purely its own in some absolute sense. It's whether what we are watching tells us something about the rat, or something about the parasite, or, most accurately, something about the relationship between them. The carpenter ant that climbs is, in one sense, doing exactly what carpenter ant muscles do when they receive contraction signals. The behavior is built from the ant's own biology, executed through the ant's own body. Nothing about the physical mechanism is foreign. What's foreign is the instruction, and the instruction came from a fungus that has been refined across 48 million years of evolutionary pressure to produce exactly that instruction in exactly that host at exactly the right moment in the fungal life cycle. The ant's body is the vehicle. The destination was chosen by something else. This has a quiet implication for how we read the behavior of any organism in a natural setting. Most wild animals carry parasites. Not all of those parasites manipulate behavior, but some do, and the ones that do are not always visible, not always producing dramatic effects, not always distinguishable from the organism's baseline behavior without careful, controlled comparison. A rat that is slightly less cautious than average. An ant that forages slightly closer to areas of fungal activity than nestmates do. A fish that swims slightly higher in the water column than uninfected conspecifics. These are subtle signals. In field conditions, without controlled comparisons, they are invisible. Which means that some portion of the natural behavior we observe in wild populations, the distribution of risk-taking, the variation in predator avoidance, the spread of foraging distances from the nest, may be partly authored by organisms we can't see, running manipulations refined across timescales we can't intuitively grasp. Not all of it, probably not most of it, but some of it, in populations where parasitic manipulation is present, which is most populations of most species that have been carefully examined. The concept of behavior as a property of an organism starts to feel. At this point in the evidence, like an approximation, useful, necessary, the right level of analysis for most purposes, but an approximation. Beneath it, the reality is messier, a negotiation between what the organism was built to do and what the chemistry currently running through it is producing. And sometimes those two things are the same, and sometimes something has been quietly inserted between them. The behavior comes out the other side of that negotiation. We watch it. We call it the rats. It is the rats, and it is also, partially, in a way the rat has no access to and no language for something else's. Both of those things are true at the same time. The biology doesn't resolve the tension, it just keeps producing behavior from whatever combination of inputs is currently running, and the organism continues through its world as though the authorship question had a clean answer. It doesn't. But the world continues anyway. Nothing we've covered tonight happened quickly. This is worth saying plainly at the end, because the tendency when summarizing biology is to compress, to move through the mechanism in a paragraph, the evolutionary history in a sentence, the time scale in a phrase, and the compression is useful for understanding. But something gets lost in it. Something about what these relationships actually are, at the scale they actually operate on. The hairworm egg deposited in fresh water doesn't hatch into an organism with a plan. It hatches into a larva following chemical gradients, toward light, toward whatever conditions its biology is calibrated to seek, with no destination in mind because mind is not a thing it has. The larva that finds a suitable host begins the process of becoming something large enough to matter. This takes months. The worm grows, the cricket feeds more than usual, running a metabolic deficit. It doesn't register as deficit. The coiling continues in the dark interior of the hemocoel, slow and patient in the way that only things without patience can be patient. Weeks pass, then more weeks. The Toxoplasma cyst in a rat's amygdala was deposited there during an acute infection that the rat's immune system eventually controlled. The control didn't clear the parasite, it pushed it into a slow form, walled it, reduced its metabolic signature to something the immune surveillance could monitor without fully mobilizing against. The cyst settled. Then it sat. Days accumulated into weeks, weeks into months. The rat went about its life, foraging, avoiding predators with a response that was slightly less sharp than it would have been, sleeping in its burrow, doing the things rats do. While inside the amygdala tissue, bradyzoites divided slowly inside their walled enclosure, and the local dopamine chemistry ran at its slightly modified level, and nothing about any of this announced itself. The manipulation was not an event, it was a condition. A persistent, quiet, ongoing alteration in what the rat's chemistry produced, running continuously for the remainder of the rat's life. The amber ant bit down 48 million years ago. I keep returning to this number because I don't think it's possible to actually hold it. Not really. Not in any felt sense. 48 million years is not a long time the way a long afternoon is a long time. It's a span that contains the entire evolutionary history of most modern mammal orders. It contains the diversification of grasses, the emergence of modern bird families, geological events that reshaped coastlines and river systems, and the distribution of habitats across continents. Inside all of that time, Ophiocordyceps and carpenter ants were doing what they're doing now. The same height, the same humidity, the same mandible grip on the same leaf vein geometry. The relationship was already old when the first human ancestors were still millions of years away from existing. It's late, or early. The distinction has stopped mattering. I started writing this without sleep, and I'm finishing it in the same condition, which means the adenosine has been accumulating for longer than I want to calculate. The irony of spending this many hours immersed in the biology of organisms that alter nervous system chemistry is not lost on me. My own nervous system has been running on blocked receptors and stubbornness, which is a less elegant manipulation strategy than anything we've discussed tonight. But here we are. What I keep thinking, in the particular soft-edged way that very tired thinking goes, is that the slowness is the part that matters most. Not the mechanism, not the molecular precision, the time. The time it took for a compound to be refined into a shape a receptor couldn't refuse. The time it took for a behavioral alteration to be calibrated to the minimum effective dose. The time it took for a fungus to learn, in the only way evolution learns, through the differential survival of everything that tried, exactly where a leaf vein needed to be. You're lying somewhere right now, or close to it. The room you're in has a temperature your body is making small continuous adjustments to maintain against. Your immune system is surveilling tissue you'll never feel being surveilled. The adenosine in your neural tissue has been building since you woke this morning, and the signal it's producing, the slow chemical accumulation of a day's wakefulness pressing gently against the receptors that will eventually open and let sleep through. That signal is real, and it is patient, and it has been waiting without urgency for you to stop blocking it. The biology doesn't rush. Somewhere in a tropical forest, an ant is walking a familiar foraging route through territory its colony has used for generations, a route that skirts by accumulated behavioral learning the areas where the forest floor is dangerous. The avoidance wasn't taught, it was selected for. The ants that didn't skirt those areas didn't return, and over time the route shifted without anyone deciding to shift it. The forest floor below a mature Ophiocordyceps fruiting body is releasing spores into air that move slowly through the understory. Most of the spores land nowhere useful. Some land on the path. The ant continues along the route that learned, across many ant generations, to stay away from the path. This time, or some future time that is indistinguishable from this one, the root and the spores will coincide. The coincidence is all the relationship requires. A rat in a field somewhere is moving through its night, nose processing the chemical landscape of the grass and soil and air. Prey, competitors, environmental signals, the faint trace of a predator scent that should be redirecting it and isn't quite, not fully, not with the intensity that the uninfected amygdala would produce. The rat doesn't know this. It moves through the night the way it always moves. The world it's navigating feels, from inside its experience of it, like the world as it is. The chemical alteration running in its amygdala tissue is not experienced as alteration. It's just what the night feels like, from inside a rat that has never known any other version of its own chemistry. A snail is regrowing an eye stalk in the damp vegetation near a stream somewhere in Central Europe. The cellular repair is slow. Tissue regeneration always is, the biology of rebuilding a complex structure from undifferentiated cells proceeding at its own pace, without reference to urgency. The sporocysts that migrated into the last eye stalk are gone now, carried away in a bird's digestive system, completing a life cycle that required exactly this sequence of improbable events to proceed. The snail doesn't register the loss. It grazes. The new eye stalk grows. All of this is happening right now while you're here, while the weight behind your eyes is doing what weight does when it's been building all day. The biology doesn't need your attention to continue. It was running before this video started, and it will run after it ends, in the forests and fields and streams and neural tissue of animals that have never heard of any of this, carrying relationships refined across timescales that make tonight feel very small and very quiet. That's not a diminishment. It's just the scale of what's alive. If something in this kept you company tonight, through the hairworm's geometry, the amber ant's grip, the quiet arithmetic of a third of all humans carrying something they'll never feel, then this space did what it was made to do. The sleepy biologist exists because people keep returning to it. And if you're one of them, the like button is a small way of telling the algorithm this kind of content is worth finding. Subscribing means the next one arrives without you having to go looking for it. Channel membership keeps this space running at the pace it needs to run, unhurried, long form, without the pressure to be anything other than what it is. If something tonight moved you enough to leave a word in the comments, a location, a thought, a question that opened while you were listening, those accumulate into something that feels, from this side of the screen, like a small and genuine community of people who find rest in the same strange places. The next video is already forming. For now, the adenosine is waiting. Let it through.